And thank you so much for inviting me. It's a pleasure and an honor to be here. I'm amazed that there are so many of you. I was thinking, I can't think of any other country, apart from the giant countries, that would have a meeting of mycologists with so many people. We certainly don't have this many in the UK. It's wonderful. You will have noticed that actually I'm not speaking Norwegian because sadly the only language I can manage is English, and I apologize for that. So I hope that most of you can understand somewhat. So far, we've heard mostly about fruit bodies, I guess, but I'm going to talk about what I think is the main part of the fungus, the part that's hidden in whatever the fungus is growing on, and that's the mycelium. That's what I'll talk about today and tomorrow. Now, fungi rarely exist alone, and certainly when Basidiomycetes meet, they fight. Actually, I'm so interested in fighting fungi, this probably tells you a lot about my own character, which... Anyway, just to get you into the swing of things, this is a tray of soil, straight from the woodland, about 24 by 24 centimetres. Blocks of wood, this one was colonised by Phanerochaete velutina, this one by the sulfur tuft fungus, Hypholoma fasciculari. They grew across the surface of the soil, and they met about there. And then they fought. And you can see that Phanerochaete is winning. I mustn't keep walking away from here. I can't turn the slides. Now, we can see these interactions not just on the surface of soil, but also in wood. This happens to be a beech Fagus sylvatica carved into a bowl, but you can see all of these wonderful lines, interaction zone lines. And these lines where battles are taking place. They demarcate individuals of the same species. So like you and I can recognize each other, fungi can recognize each other too. So these are individuals here of the same species. These lines also demarcate individuals of different species. So there's a different fungus in there compared with there. They're a bit like us, that we have fences or walls or hedges around our homes, around our territory, and they do that too. And so any fungus protected by its wall around it can then use the wood that's in there, the food that's in there, at its leisure, so to speak. Now, fungi can fight in lots of different ways. They have different modes of attack and defense. They can fight at a distance. They can produce diffusible substances, gaseous substances, which always makes me think of the dreadful trench warfare in the First World War. And you can see that these fungi, they've actually not met. They never do meet because they produce these substances which act at a distance on their opponent. They can fight in other ways too. There's hyphal interference, which is a sort of mycoparasitism. You can see that here. This is Phlebia gigantea, and this is Heterobasidion. Phlebia has touched Heterobasidion, and look at the state of the contents of that cell. That's been killed. And this is the basis of the biological control of Heterobasidion. Then there's the more usual forms of mycoparasitism where hyphae perhaps coil around other hyphae and maybe penetrate inside. And some fungi do this as their only way of gaining food. Wood decay fungi often do this to get territory from another fungus. So they will parasitize it, get all of the territory occupied by that fungus, start rotting the wood, and then start fighting everything around them using different mechanisms. And then the last way of fighting that I put here is what we've called gross mycelial contact. You've seen an example of that. This is a very rough and ready catch-all term because the truth is we don't understand many of the mechanisms yet, but there are very many. And so there will be more categories later on, I dare say, probably after I've retired. But I'll tell you what, though, in the UK, you can work as long as you like. You don't have to retire at 70 like you do here. So we may find out more yet. When fungi meet, all sorts of changes occur. And you can see these most clearly in agar plates. Now, our early work was done on artificial agar media, like you can see here, but now we rarely do this because it's just so far from the real world. We usually look at interactions in trays of soil or in blocks of wood. But obviously, from the point of view of seeing things, this is the easiest way to do it. So I put up quite a few Petri dish pictures. You can see that when mycelium meet, all sorts of morphological changes occur. Sometimes they become aggregated into cords. This often happens if they produced a horrible zone of chemicals which has killed their opponent. It's a way of getting across. Sometimes they produce chemicals which are colored, that you can see here staining the agar, and here. There's another nice example of antagonism at a distance. These fungi never meet. And then there are masses or barrages of mycelium where the attacks and defenses are going on. Now, I'm only going to touch upon this briefly. As you would guess, when fungi fight, all sorts of changes occur. The way that genes are expressed change. And we looked at this initially about 10 years ago. I'm only going to talk about this briefly because actually things have moved on since then. But we had Trametes versicolor, the turkey tail fungus, fighting against other fungi. And this was done in Agar. It fought against Sterium gaussipartum and Trametes beatsterium. It fought against Jacandra, and that's a draw. And it fought against the sulfur-tough fungus Hypholoma and Hypholoma 1. So win, lose, and draw. And we looked at gene expression by using subtractive cDNA libraries. The point is, some genes were upregulated, some were downregulated, some were found in all interactions, some were found in 2 of them, and some were found just in 1. As I've already mentioned, they produce volatile chemicals, gases in the air, to attack their opponents. And we can see this here. We put a block of wood colonized by one fungus against a block colonized by another fungus and let them fight. And then we measured the gases that are produced in the air above them. And using a machine called a gas chromatograph mass spectrometer, we can see the gases that are produced. This is for Hypholoma against itself, the red trace. Each one of these little peaks is a different gas, a different chemical. And you can see when it's on its own, there's 3 things. With Resinicium bicolor, only 1 thing, but put the 2 together and masses and masses and masses of different volatiles are produced. Actually, one of them decreases, but all of these others are produced or increase. There are all sorts of exciting things going on. Now you can look at the effects of these volatile compounds by making fungi fight in the top of a big Petri dish like this. There's 2 fungi in wood blocks fighting away there. You can put another one on the bottom, let it grow out, and see how quickly or how slowly it grows when there are fighting fungi above it. And indeed, very often, these fighting fungi will inhibit the thing below, or sometimes they might stimulate it. These fungi produce all sorts of different chemicals, I should have said. And if you know about plants being eaten by insects, lots of the chemicals that fungi produce when they fight are the same as what plants produce when they're being eaten by aphids or whatever. Anyway, so we know that when fungi fight, they inhibit other things. Now, I had a student who did this work, and I always tell my students, read everything that I've written about and that everybody else has written on this subject. Think about it, but don't believe it until you're actually convinced. And the same is true with anything else I say to you. And this particular student thought, okay, when fungi fight in wood like this, they produce lots of different volatiles. But when they fight growing across soil, they produce different compounds. Now, she'd done this experiment here with the fungi fighting in wood, and she said, what I'm going to do next is I'm going to let fungi fight on soil, and I'll put that in the top and have something else in the bottom and see the effect. And I said, well, do you know, there's this thing that was discovered in the 1600s called gravity. And surely the soil will fall down. Anyway, she thought, okay, went away. And a few months later, she came back. She said, I've done that experiment. And I said, which experiment? She said, oh, the one with the soil in the top of the tray. And I thought to myself, oh, well, maybe we don't have gravity in Cardiff. But it turns out if you wet the soil and you squash it up, it does stick in the top for long enough for the experiment to take place. So the moral of that is to any students in here, don't believe what your supervisor says. Now, you will have guessed that fighting is an expensive occupation. It's energetically expensive because the fungus has to produce all sorts of different compounds, do all sorts of different processes to win these battles or to survive. And how does it pay for that? Well, sometimes it pays by stopping growing, but very often it pays by doing more decomposition, decaying more quickly. And we can see this with this data here. When you've got, well, let's say that the line across the middle is the situation if a fungus or if two fungi were growing on their own. The bars above are when more decay has occurred when fungi are fighting. Bars below are when decay has got slower. And yes, sometimes decay gets slower when fungi fight, but usually it gets quicker. And that is showing you that fungi are paying for these battles by breaking down wood more quickly. They've got to get the energy and nutrients from somewhere. And we looked at that by looking at weight loss of wood. Another way you could do it was by measuring CO2 evolving into the air when fungi fight. They're breathing like us. We breathe out CO2. They respire CO2 as well. When the fungi are growing on their own, over 84 days, not much change in CO2, but when they come together and fight, you can see that very quickly they respire more rapidly, as would you or I if we were fighting. And at this stage, I ought to point out that I am a black belt in karate, so please behave. Well, you will have gathered from what I've said so far that there are different outcomes to these battles. Sometimes there's deadlock or a draw when neither fungus gains any extra territory. Sometimes one fungus will replace another. Sometimes a fungus will start to replace an opponent, but suddenly it stops. Well, whether it runs out of steam or whether that other opponent has suddenly been able to turn on some defensive machinery, we don't know yet. Also, there's this strange situation which sometimes occurs called mutual replacement, where fungus A replaces fungus B in one region and fungus B replaces fungus A elsewhere. And you can get a hint of this in this tray of soil. This fungus is growing out, but you can see that the cords are just gradually growing over this other fungus here. Not only do they grow over, but if I showed you a later picture, you'll see that they have killed the mycelium beneath. On the other hand, this one round here is coming around in this direction. It always reminds me of when I was a child and they used to have cavalry battles on the television, and one army would go this way and the other army would go the other way. I've shown you so far static pictures at one instant in time, but of course these battles take place continuously. Now, there's a little bit of time-lapse here, and it's probably not worth waiting for, actually. This is the very first bit of time-lapse photography that we saw of these interactions. This is Trametes versicolor growing on agar plate. This is Daldenia concentrica, the ascomycete. Trametes is exuding lots of enzymes. It's destroying Daldenia. The mast mycelium of Trametes is growing through the region where it's killed Daldenia. It's producing more enzymes and carrying on growing. And actually, it's getting up such a head of steam now that it's growing through Daldenia even quicker than those enzymes are killing the Daldenia below. And if we waited a bit longer, this fungus will completely replace that one. We've got another example here. That was in agar, but as you said, I'm not so keen on agar. Is it going? Yes, it is. This is growing across soil. It's Phanerochaete velutina growing from a twig. This is Hypholoma fasciculare, the sulfative fungus. In a minute, you'll actually see Phanerochaete coming around the top. Well, they've just about met, and oh, Hypholoma is starting to replace Phanerochaete. That was a surprise. In all the other times I'd done it before this, it was the other way round. And I was wondering, will Hypholoma this time win, despite all this Phanerochaete velutina? But unfortunately, the person who was filming it got bored and stopped, and I never found out. So, obviously, Different fungi are better fighters than others. They replace at different rates. And we thought we'd have a look at this. And again, we colonized wood blocks with different fungi. Imagine my fist of the wood blocks. We put the wood blocks together. And then over 84 days, every week, we took these blocks apart and took little chips of colonized wood out to see what fungus was there. And this is what we found. So we're always considering this fungus, Voluminia comedens, an early colonizer of attached oak branches. So this is time along here. This is how time changes, what happens. This is the amount of territory occupied by each fungus. So at the start, Voluminia occupies half of the territory. It's in one of these wood blocks. Trametes is in the other wood block, so it occupies half. After the first week, nothing happened. After the second week, nothing happened. After the third week, after the fourth week, the student was getting frantic by now, thinking this is a total waste of time. But then suddenly, replacement. And that happened very quickly. So maybe that fungus, maybe Trametes takes a while to get together its arsenal of compounds to beat Voluminia. In this combination here, Voluminia, Sterium, well, no change all along. Actually, Sterium is a great defender. It's a very poor attacker, but a great defender. So no change. This one, you see the Jacandra eventually takes over. Quicker still, Hypholoma, and look at this, the great Phanerochaete. Within 2 weeks, it's completely replaced its opponent. So the great fighters replace more quickly than the less good ones. And actually, you can see a sort of a hierarchy of fighting ability amongst fungi. And this reminds me always of a sports league. I'm afraid, I'm sorry, I don't know much about sport in Norway. In some countries that I go to, if I would learn a football analogy. So when I was in Germany recently, I learned the Bundesliga and related all these fungi to the different teams in the Bundesliga, but I can't do that. So we'll have to stick perhaps with UK football teams. So some fungi at the top of the league, they beat most other things. So that's like Liverpool and Man United. Now, actually, I come from a small city called Worcester. We don't even have a football team that's in the Premier Division. So we'd be somewhere down here. So all these teams would mostly beat us. But let me take you back to 1959, when in the FA Cup, Worcester City, down here, beat Liverpool. That happens with fungi too sometimes, although these are the best ones win most of the time. Sometimes there'll be a giant killer down here that occasionally might beat another one. I was talking in Zurich a couple of weeks ago, and you'll see that this is Armillaria gallica at the bottom. There were a whole group of people there who worked on Armillaria, and they were very upset that Armillaria is such a pathetic fighter. So I hope there's no Armillaria fans in here. Anyway, the outcome of these battles varies depending on the conditions. So if we've got two fungi which maybe grow in soil and they also grow in wood, the outcomes of the battles may be different in soil and wood, and certainly they're often different in agar. If the climate changes, the temperature, the moisture, the gaseous regime, that affects the outcome. So with climate change, we're going to see different communities developing. How much food there is alters the outcome of battles. Whether there are other microbes or other fungi present has an effect. And so does grazing invertebrates. So, as I mentioned, invertebrates can affect interactions. This is Hypholoma, this is Phanerochaete, and they meet. And then we added some calembulus, some springtails. Sorry, I don't know the Norwegian word. Anyway, the point is that these little creatures eat fungi. So what happens? We've gone very colourful here. Now, what we're seeing is those same trays of soil, but 33 days later. So this is obviously the situation now, and these are where the cords are. They're coloured in now. These are the ungrazed ones. These are the ones where the little beasties were there eating away. And what the colour stands for is the blue colour shows where mycelium has disappeared in the 33 days from when the previous photos were taken. So this is now, this shows you what happened compared with 33 days ago. So in the ungrazed ones, actually lots of mycelium has died. The red shows you where there's been, and the yellowy orange shows where there's been new growth. So where there's no grazing, the fungus grows all over the soil tray, no problem, because it's not been eaten. But here where the grazers are, doesn't grow very much. Few little patches there, look. But elsewhere, it's been nibbled, 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 eaten away, no growth. But there's one huge surprise which we never predicted, and that is that the Fungi, when it was being grazed, could grow massively over the hypholoma and start to replace it. Look at all this mycelium here compared with there. You would never have guessed that. That's sort of counterintuitive, isn't it? But I think one of the reasons is that the Calembula don't like eating hypholoma because actually it's toxic and kills them, so you can understand that. And so they actually don't really want to go on there, so the Phanerochaete is safe to grow over there. It won't be eaten either. And just to give you a few examples of how they can completely affect the outcome of competition. This is Hypholoma fasciculare fighting against Resinicium. Resinicium is winning. This is the same situation with Calembula. Calembula eat all of these hyphae, and actually neither fungus wins. They keep their own territory. Here's another example, Hypholoma against Resinicium. Resinicium winning. Put in nematodes, these little wiggly tiddly worms, which insert their little stylets into the hyphae to feed. Complete change, and the Hypholoma starts to win. With wood lice, oniscus, isopods, these things feed very delicately. They nibble, nibble, nibble hyphal tips. The isopods, the wood lice, they graze like I mow the grass. So that's not in nice straight lines. I mow the grass like this, and that's how they feed. They have huge effects. Resinicium was beating Phanerochaete. In come the grazers, yum, 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 yum. Complete change, and this one wins. So if you start to think about these fungi in the field, all these things are happening in the field. Now, so far, I've told you a very simple story. We've always had just two fungi fighting, but that doesn't happen very often in the real world. We can see here, there are lots of fungi fighting each other. This one's got 1, 2, 3, 4, 5, 6 on the boundaries we can see. If you imagine this as a whole trunk of tree, there are lots of interactions going on simultaneously. So we thought we ought to start to look at the more complicated situation. Now, could we predict from everything we've done so far what happens? Who's going to win? If fungus A beats fungus B, and then fungus B beats fungus C, we know that fungus A would always win everything. But does that happen in the real world? Well, we get these strange situations. Fungus A can beat fungus B can beat fungus C, but fungus C can beat fungus A. They use different mechanisms, so then you can't predict. There's an example here. If you look at the tip of the arrows, they're the winners. Sathurella beats Phallus. Megacalibia beats Sathurella, Phallus beats Megacalibia. And an even more complicated situation too. These are some of the main players in the decay of beach logs, Fagus sylvatica. So here's the great one, the Liverpool or Man United, Phanerochaete velutina, wins nearly everything. You can see the arrowheads, the dark colours when it wins quickly, light colours when it wins slowly. Beats everything almost, but not Sterium hirsutum. That's the great defender. However, look, Hypholoma beats Sterium, and Phanerochaete has beaten Hypholoma. So it's not simple. It was about 20 years ago when we thought, first, we ought to look at this more complicated situation, and actually, we did one experiment and then stopped because it was a bit complicated. Anyway, soil tray again, Phanerochaete grown from a small wood block, and actually it grew out fairly circularly, and then we put straight onto the edge of the mycelium 3 blocks of wood colonized by different fungi. And if this Phanerochaete met one of these on its own, it would always win, always. So you might think that when it gets all 3, it'll just start to fight all 3 of them straight away, but that's what it never did that. It always beat the weakest fungus first. In this case, Kretschmeria, the Deosta, the Ascomycete. So fungi are bullies, basically. They pick on the weakest, they beat the weakest, and then move on to the next. However, that was still a bit of a simple situation. So we went a few years ago to a bigger situation. 3 things competing, followed them all with time. They're all in different orders. So these ones are Phanerochaete, Hypholoma, Sterum, Hirsutum, but in different orders. And let's just have a little look to see how order in which they meet each other affects the outcome. And that's illustrated here pictorially. So if you have Hypholoma on one end, Phanerochaete on the other end, and something else in the middle, after 84 days, Phanerochaete has nearly always won the whole lot of the territory. On the other hand, if you keep Phanerochaete on the end, but put Hypholoma next to it, and the other things beyond Hypholoma, it's different. Hypholoma does so much better. So it really matters what that fungus is sat next to, who it fights first. Well, let's get a little bit more complicated still. This is getting like branches and logs with lots of fungi in the community. And I thought back to my childhood. Remember those dreadful Rubiks Cubes? I could never do Rubiks Cube. I hated Rubiks Cubes. But they're quite good when it comes to fungi. So these are Rubiks Cubes. Yellow is one fungus, green another, blue another, randomly assorted in that cube, but they were never allowed to have two blues together. That was the one thing that we set up. Then we had a different situation where we had a sort of a wall with all the blues. Same amount of blues here as in here. These ones were just random. So this is that situation. And the wall is of a very weak fungus, in this case, Voluminia comedens, that we get in the canopy. And we'll just concentrate on these 2 here. This is the Rubiks Cube. This is with the wall. In the Rubiks Cube, Voluminia does terribly. In the wall, it does much better. So that accounts for why it really does quite well in attached branches, where it forms these great long individuals as a wall. If it was dotted about here and there, it would have no chance. But as a wall, it does pretty well. I'm going to very quickly, I promise, tell you about what happens then when you look at the gases, the volatiles that they produce when they're growing together. So we looked at the gases produced, the volatiles produced in that situation and in that situation, and indeed when the fungi were growing on their own. This is when they're all growing on their own. This is the Rubiks Cube with random placement. After 1 day, after 28 days, this is the wall after 1 day and after 28 days. And the fact that all these little blocks are separate shows you that in all of these conditions, they produce different gases. Now, let me tell you this. We paired them separately, that against that against that, and that one against that, and measured the volatiles they produced. And then if you made a list of everything that was all of these gases that were made, if you made a list, you'd think that's it, that's all that those fungi can ever make. You'd be wrong. Put them together like this, and they made more and different ones. How can that be? They've got these emergent properties. And we looked at enzymes in these situations. And again, I'm not going to go through the data. They were different in the walls compared with the Rubiks Cube. There were always more enzymes when Voluminia was a wall. That was the volatiles. This is the enzymes. There was always more when they were a wall than a Rubiks Cube. We then looked at all of the metabolites, everything that the fungus produced, similar patterns to on that map before. So I just quickly flashed through that because I wanted to get out into the field. You probably want to get out into the field by now, or at least a coffee. Okay, so everything I've told you so far, we've done in the laboratory. But does this bear any resemblance to the real world? Does the fighting ability that we see in the laboratory time with what goes on in the field? If we've got one species in wood in the field and then another one, do the communities that follow alter? So does species A attract different fungi than species B? So we tried to answer those questions in a field experiment. So we took discs of wood, colonized them with a single fungus, well, 9 single fungi. It happened to be Hypoxon fragiforme in this case. We put all these blocks colonized by, singly by different fungi out in the wood and collected them after 6, 12, 18, 24 months. We found nearly all of them. I do emphasize nearly all of them because there were lots of Hypholoma discs that we didn't find. And we couldn't understand this because we randomly positioned them on a grid in the wood. We knew where they were from what we'd written down, but no human could have known other than us with the list. So how come Hypholoma kept disappearing? Where did it go? Dotted all around this woodland, they disappeared. And I'm pretty certain that it's badgers. Because we discovered that we were actually on a badger latrine site, which was a bit unfortunate. And I think that badgers either like Hypholoma very much or else they dislike it intensely. Anyway, they'd moved it, so we didn't get half of our Hypholomas back. But those ones we did get back, we re-isolated into agar culture, and for the one set, we did molecular sequencing to see what was there now. So what did we find? So this is Voluminia, and then the 2 Ascomycetes. These are 3 intermediate phytos from the lab, Trametes, Sterium, Jacandra, and these are the great phytos, Hypholoma and Phanerochaete. The red color means they survived for 6 months. Green color survived for 12 months. These early colonizers were not there after that. Blue color, they've been there for 18 months. Trametes is still there, as is Sterium, Hirsutum, and Jacandra and the others. Purple, after 24 months. Trametes is gone. Sterium, the great defender, is there. The great attackers, Hypholoma and Phanerochaete, lots of them still there. Okay, so that fits with what we found in the lab, so that's nice. But did they affect the communities of fungi which followed them? And the answer to that is yes. So if the ovals overlap, that means there's no difference. So this was uncolonized wood, and the other 2 colors represent the Ascomycetes. These were all Basidiomycetes to start with. This is Sterium hirsutum, the great defender. So the Ascomycetes and uncolonized wood have different communities coming in after them compared with the Basidiomycetes. And the great defender, Sterium hirsutum, has different communities coming in than the other Basidios. So what are the fungi that come in after these earlier colonizers? If you're interested in Ascomycetes, look at the top. If you're interested in Basidiomycetes, look at the bottom. If you're not interested in either, why are you here? Anyway, the point is, these were the controls with nothing in originally. These were the Ascomycetes to start with: Voluminia, Trametes, Sterium, Jacandra, Hypholoma. And the colors represent the things which came in later on. And the point to note is that different starting fungi attract different fungal communities later on. Now, this is just to sum up. You'll be relieved to know, because I'm sure lots of you are waiting for coffee, or in my case, tea. I'm British. Fungal phytes alter many things. We've already seen they alter gene expression, volatiles, enzyme activities. They also actually alter the way mycelia search for new food supplies. And I'll talk about mycelia searching tomorrow. I haven't talked about how the phytes alter this. They also alter nutrient distribution and release. So here's it doesn't matter that there's several blocks of wood, but this is Hypholoma, Phanerochaete. We added phosphorus 32, radio-labeled phosphorus to there, or to to neither or both. And what you see to start with is when they, at the start of a battle, when they meet, they shunt their phosphorus to the battle line. But then, when, in this case, Phanerochaete beats Hypholoma, Hypholoma madly moves all of its phosphorus as far away from the opponent as it can. And the best unpublished piece of work, the piece of work that I've never written up, is another experiment which shows when nutrients are released. Fungi, or decomposer fungi, are inherently greedy. Wood decay fungi find it difficult to get hold of nitrogen and phosphorus, and when they've got it, they want to keep it. But we know that they release it, because otherwise how would plants carry on growing? And so I think that they release when they're grazed by invertebrates and when they fight with an opponent. So we set up an experiment where Beneath here, we had the ability to catch liquid coming through different regions. So we had one region there, another region in the zone line there, and then another one there. We added a P32, and then we let them fight, and we watered them as if it was raining and caught what came through. So I was expecting that masses of P32 would come through there because they're fighting, and it's going to be lost. And yes, that happened. Hurrah! However, as so often is the case with me, I didn't get the whole story right, because strangely, lots of P32 came through from there and there too. So the whole of these mycelia become leaky for nutrients when they fight. Decay rate increases when they fight, usually, as I've mentioned, and the communities of fungi change, and hence decay rate also changes.